Morning. This is kind of a continuation of this whole new idea of to look for cancer in the blood. And I've had so many questions, so I figured I start off the morning by making this video explaining things a little bit and trying to clarify things and not assuming that everybody has to uh, kind of read everything and waste your time. So before we talk about finding cancer DNA in the blood, we first need to understand what DNA is and how it gets into our cells in the first place. So, every one of us started as one single cell, an egg or ovum that came from mom, sperm that came from dad. The egg carried the DNA from mom, and the sperm carried the DNA from dad. When the egg and the sperm are joined together, now we have a combined DNA of mom and dad together. So, in simple terms, about half of our DNA comes from our mother and the other half comes from our father. So, that became our own unique genetic instruction book, half mom, half dad. And the basics of it is that the first cell, that's half mom, half dad, is divided and then it becomes two, then it becomes four, then it becomes eight, and it goes on and on and on. And this continued until we eventually became trillions of cells, and maybe a little bit more. Each time a cell divided, it copied its DNA and passed that DNA to the new cells. So the information kept on going and going and going. That's why every cell in your body today contains DNA. So, what exactly is DNA? Going back to med school, I think of DNA as the instruction manual inside my cells. It contains the information that helps me uh, to know how to grow, how to function, how to repair myself, specifically speaking about the cell, and how to make new cells uh, in addition to getting rid of the old cells. So, the only problem or the positive aspect of it is that our cells don't live forever. It's a good thing. Nobody wants to live forever. So every day, cells throughout our bodies get old, become damaged, and they die. Your body's constantly replacing those cells. When some of those cells die and break apart, tiny fragments of their DNA are released. Some of those DNA fragments can be found circulating in the blood. You draw the blood, you look for those broken down, regular run of the mill dead cell DNA. And in science, they call that cell free DNA or cfDNA, regular DNA. And this is important to understand. So, having cell free DNA in your bloodstream, which can be tested, is normal. So, that's not a concern. A lot of my patients ask me, I have cell-free DNA in based on the very expensive blood test I did overseas, and what are your thoughts? And I always tell them healthy people have cell uh, free DNA circulating in the blood, and the labs that test for it basically are confirming the fact that you don't live forever. That's it, and we move on from that. So, if a laboratory finds a DNA fragment in your bloodstream, that by itself does not mean anything. We expect DNA fragments to be there, so that's that. Now, let's bring the cancer part to the story, the part we care about. Cancer develops from our own cells. A normal cell can acquire a change that allows it to grow and divide when it shouldn't. But cancer cells don't live forever either. Cancer cells can die and they break apart. And when they do, they can also release fragments of their DNA into the bloodstream. So imagine I draw a tube of your blood, and floating inside that tube, I will find millions of tiny fragments of DNA. Most of them are cell free DNA that doesn't come from a tumor, but in someone with cancer, some of those fragments may have been released by cancer cells, and therefore that cancer derived portion is called circulating tumor DNA. Makes sense. All right. Now, the term liquid biopsy starts to make more sense. Liquid means your blood, biopsy means I'm drawing your blood. Why they make it sound so fancy, I really don't know. But it's not a big deal. We got it. So traditionally, if we want to learn about a tumor, we may need to put a needle into it or remove a piece of it. That is called a biopsy of that tumor. What a liquid biopsy then does is it takes a tube of blood, it analyzes the material in that tube of blood. The material that we're focused on are the DNA fragments, and we're essentially asking that tube, do any of these DNA fragments contain clues suggesting that they came from a cancer? So different tests can look for different clues. Some look for what are called mutations, changes in the DNA associated with cancer. So examine DNA that we could understand. Some examine the actual DNA methylation pattern, which basically refers to the fact that the chemical changes on the DNA that can differ from normal cells. So it allows us to compare the two. And a newer approach can even examine the size and the pattern of those DNA fragments. It's pretty darn amazing. So now we get into the question that I think is most important. What happens if someone has never been diagnosed with cancer, like my son, and he feels perfectly healthy, works out, goes to the gym, starts college, and the blood test that dad knows a whole lot about comes back and it says that they detected a signal that appears to be associated with cancer. Does that actually mean my son has cancer? The answer is really no. It basically means that the test detected a signal that may deserve further investigation. I love my son. I don't want him to have cancer. So perhaps cancer really is present and is still very small, and perhaps it doesn't or yet hasn't caused any symptoms. But blood tests are not perfect. Abnormal signals in all these new artificially intelligent testing methodology can occur. And so this is kind of like to, uh, in a sense, thinking of a smoke alarm. If your smoke alarm suddenly goes off, does that prove that your house is on fire? No. Maybe there is a fire. Maybe it's smoke from the kitchen. Maybe something else triggered the alarm. But you certainly don't ignore it. You look for the source and you look and see why the heck there's fire. So a positive cancer related blood test can be similar, and it may be telling us that something could be here. Now, we need to investigate. Depending on the situation, that investigation might be doing a CAT scan or an MRI or a PET scan or a mammogram or an ultrasound or a colonoscopy or an endoscopy or another appropriate test on my son because the test came back positive. And if eventually we end up identifying a suspicious mass on one of these tests, then it makes perfect sense to do a biopsy of the bump or the mass that shows up somewhere. So uh, that's basically, in a nutshell, meaning doing this test, if it does come back positive, will tell me to do additional testing, find something, and biopsy it. And biopsying it then may tell me it was nothing or it was something and we caught it early. I want my son to go to school and graduate from Texas Tech and do great. So, number one, normal cells die every day and they release fragments of DNA. We got that. DNA fragments circulate freely in the body and they're called cell-free DNA. We got that. Having cell-free DNA or regular dead DNA of our stomach, of our blood is normal. We got that. Number two, cancer cells also release DNA fragments, and the cancer-derived portion of cell is called circulating tumor DNA. Cancer DNA, tumor DNA. We got that. And number three, the term liquid biopsy then is really allowing us to examine the material in the blood for molecular clues that there may be associated with cancer. But again, positive cancer related signal does not automatically mean you have cancer. It means we may have found an important clue. And a next job of the medical team is going to be to determine whether the clue came from something legitimate or something bogus, which is what I call an incident or an incidentaloma. I'm Dr. Alex Eson, about to start my day, heading to clinic. I just wanted to put this out there. I apologize, it's a little bit long, but I know it answered 1,500 emails, and I hope it was useful to you. Have a blessed day, and we see each other soon.